So in today's special lecture, we have Dr. Terry Jernigan. She's, I think, the third in the lecture series now. She is a professor of cogs, psychiatry, and radiology, and is the director for the Center of Human Development at UCSD and the co-director for the ABCD Study Coordinating Center. And today she'll be talking about her career and her research. So thank you for coming.
It's my pleasure. Thank you for inviting me. Can you hear me okay? Sometimes my sound goes weird and I have to fix it.
I think it's good, yeah.
Good. Just let me know if it changes. So yes. So when I saw that you wanted to hear something about my career path, I almost blanched because I have a really, really long career. I'm really old, and so it's a long story. I'm going to try to give you the brief version of it, but I did grow up in Alabama and went off to college at the University of Texas at Austin when I was 17. And I spent 2 years in Austin, which I loved, by the way, which for me, coming from Mobile, Alabama, and not from a family with a lot of academic people in it, in fact, none, my vision of the world expanded tremendously in Austin, Texas, which is a world-class university, and it changed my life. And then I left there after 2 years and went to Southern California and resumed my education at UC Irvine. That was in, well, 2 years later, I graduated at UCI in 1973. So, long time ago. And at that time when I graduated from UCI with this very interesting education that put together a more conventional psychology degree at Austin, a wonderful one, but a kind of a conventional, traditional one. And then I ended up with in my master's degree at Irvine in this brand new university. It was really new, a few years after it was founded, I think. I mean, it's, and all of the disciplines were mixed together, and there was a lot of talk about psycho-cybernetics and things like that. It was a totally different kind of program, but the combination was fairly intoxicating, I guess. But I was really vacillating at that time between pursuing a clinical psychology PhD, a graduate program in clinical psychology, which I always kind of thought I would probably do as an undergraduate, and instead going into neurobiology. And because I had become interested in neurobiology, sort of. And but I vacillated long enough that I only managed to get one application submitted before the deadline, and that was to a very highly rated clinical psychology PhD program at UCLA. And miraculously, in my view, they admitted me and offered me a stipend, which I definitely needed at that time. So I accepted the offer, and that sort of made up my mind for me. But I spent the next 6 years, kind of, I think in retrospect, I would say kind of breaking the mold with, again, very, fortunately for me, is full support from the senior faculty in my PhD program at UCLA. I kept saying, well, can I do this instead, or can I do that instead? And they just always said, you can do that. And they made it happen for me. So I was very lucky to end up at UCLA because it was a group of people who were happy to break the mold if they thought it was a good idea. So in parallel with completing my clinical training at UCLA in clinical psychology, I arranged to get extra neurobiology training at another UC campus. I was allowed to get training in neurology at the Palo Alto VA hospital, which is quite far away from UCLA, you'll probably, where I discovered a prototype CT scanner. That really dates me, prototype CT scanner in the basement. And miraculously, again, I got permission to program the computer. Turned out I knew how to program the computer that was running the scanner, and I got permission to program the computer on it at night after the clinical schedule ended. And from that, I developed a dissertation project that sort of straddled Stanford and UCSF, even though I was a UCLA graduate student, which was permitted, using quantitative CT imaging to study normal aging. And basically during that period, I kind of fell in love with brain imaging and brain imaging research. And a then brand new branch of clinical psychology that was just kind of coming together as a formal discipline in clinical psychology called clinical neuropsychology. And I'll jump to the chase. All of us early researchers who were trained this way at this onset of clinical neuropsychology kind of all saw our field subsumed by what we now call cognitive neuroscience. So lots of changes and transitions. I remained at Stanford, based at the VA Medical Center there for my postdoctoral work, which expanded my quantitative brain imaging work to include many collaborations that allowed me to apply my methods to several neuropsychiatric conditions that were being studied there at the Stanford, Palo Alto VA and the Stanford Medical School. And in 1984, I was recruited to UCSD in psychiatry, and I was joint appointed in radiology to continue my brain imaging work, together with a number of other people who were recruited to psychiatry and radiology and neurosciences. All three departments were recruiting people at the same time. And we all sort of arrived at UCSD at the same time with kind of interest that came together in this area. So my work has consistently involved the application of brain imaging techniques in investigations of the neural bases of cognitive, social, or emotional deficits or symptoms in numerous neuropsychiatric conditions and in normal individuals across the lifespan. And as you might guess, since I've done work in so many different disorders and looking at so many different groups, and all of my work has been highly collaborative. I've always worked in large teams of researchers, never more than I do today, as you'll hear when I start to talk about my research. So for the past 15 years, since about 2005, first while I was working as a visiting professor in Copenhagen, in Denmark, and continuing when I returned to UCSD, my primary research interest has been in the origins of individual differences in behavioral phenotypes or behavioral traits and their roots in human development. And for this work, I was appointed as director of the Center for Human Development, which Christina mentioned. And then I received an appointment in the Department of Cognitive Science. So that's when I added the Department of Cognitive Science, which is now my home department, to my psychiatry and radiology appointments that I was appointed to when I came to UCSD in 1984, a really long time ago. So that's in brief, a very, very long career path. And the rest of it is kind of in my talk, especially this last 15 years. Right, so I could pause here and just ask if anybody has any questions about that, and before I start talking about the research itself.
I was curious because you said you did programming. Like, what kind of programming did you do?
That's a good question. You know, when I was at Irvine, this is one of the reasons that, you know, you think about all the things that happened that were just fortuitous, but they ended up having a profound impact on your career. You know, you'll see this when you get old, you'll recognize all those things. But at UC Irvine, the people in social, my bachelor's degree is actually in social sciences. It wasn't even in psychology, even though that's really what I trained in most of the time. But bachelor's degrees, people were supposed to do a programming class, and it was basic. So the only class I ever took in programming was basic. But I taught myself several other languages. But these are languages you guys wouldn't even recognize because they're long gone. They're Pascal and Perl. And then I taught myself Fortran. And Fortran was a very big power language at that time. It was kind of like the C or C++ now, you know, Fortran. And I taught myself to program in Fortran. And I did my initial graduate work programming what was called then a mini computer, which was the size of a closet. That was called a mini computer. It was mostly programmed with Fortran. And so I used that to do my graduate project. And then when I got to Palo Alto and I found out about this amazing machine down in the basement of the main hospital building that allowed you to actually see inside the human brain. I mean, you guys can't even imagine what it was like back then. You couldn't do this. You couldn't look inside the skull and see the human brain in 3D. But this machine did that. It was the first one. CT came before MRI. And I was actually able to program that computer. The computer that ran that scanner was the same computer I had done my graduate work on back at UCLA, my first project. And I could program it in Fortran. So that was that.
Do you still use programming like right now in your work?
I don't do programming anymore, but I work very closely with Anders Dale, who I'll talk about a little bit later in the talk. And he's younger than I am for sure, but he runs the Center for Molecular Imaging and Genetics, and we collaborate on all of the projects, as you'll see. And he still, to this day, does a lot of programming himself. And I'm envious because I loved to program when I used to do it. I play other roles now in the research, as you'll see. But I loved it. I loved programming. I remember telling people at the time that I knew women who just found that when they did certain kind of needlework or embroidery or something like that, they just found it kind of soothing and relaxing, and it made them feel. And I thought, that's what programming is like for me. I love to program. It's just like it's slow, methodical. You have a bug, you have to work it out. It's but you always get it right in the end. And I don't know why. I love to do it. So, but I don't do it anymore, I'm sorry to say. I mean, obviously I do some scripting and things like that, but not really programming.
Like SQL or R or?
Oh, well, we do a little bit of R, but mostly my postdocs and graduate students are doing that. But yes, that's the kind of stuff, that kind of scripting stuff we do. Writing scripts and programming applications and statistical applications, for example. Yeah.
Thanks.
But really honestly, if you can do it in one language, you can. I think the more important lesson is you can do it in any language.
Mm-hmm.
I think it's much easier to learn a new programming language if you know any of them than it is to learn a new language, right?
Yeah.
Much easier. It's just, you learn the syntax of the new syntax. That's all it is, really. Quickly once you start using it, especially if you switch. If you're doing them simultaneously, sometimes you get a little crosstalk, but it's easy to do. I would encourage you not to ever let that be a barrier. In a month, you'll be completely converted. Okay. All right. Any other questions? I'm not looking at the chat, so I'm not good at doing that. Let me just see if there's anything else. Yes, the language basic, that was it. Not basic as in entry level. No, the language itself was called basic. And everybody at that time knew about it anyway, knew what it was. Just it's been decades and decades and decades ago. Okay. Shall I go on to the research?
Yeah.
You want to let me share my screen, or can I?
Oh, yeah. I allowed it.
Okay, let me just do it then. Okay, can you see my title?
Yes.
That's my title for you guys. Hope I got your name right.
Yes, you did. Thank you.
Good. All right. Well, thank you for inviting me. I appreciate it. I like to start these talks by kind of saying that without much risk of controversy, I can probably safely assume that all of us want more people in our societies to thrive intellectually and creatively, and fewer people to become disengaged and possibly to fall prey to damaging dependence on substances or other habits, and for more people in our society to achieve a greater sense of well-being. But we would also probably all agree that some people learn more, create more, become more emotionally resilient, and some just have more fun than the rest of us, right? That colleague I mentioned, Anders Dale, for example, he just has more fun. But the question is why? And for answers to this question, we could probably achieve much less consensus. Is it because some people are constitutionally endowed with different personal assets, or native intelligence, motivation, optimism, capacity for pleasure? Or is it because of formative and sometimes even transformative experiences that either enhance or diminish our lives? Well, whatever the origins of these things are, I'm gonna have to make a change here because I've got the panel hiding my slides from me. I can't see them. I always have trouble when I have two things. These differences between people that I'm talking about, we call them behavioral phenotypes. But they really are just individuality. They're really just things that differentiate us from each other, personality things, habits that we have. And they can be skills like calculation or literacy, levels of knowledge or expertise, or more ineffable qualities like creativity. They can be social or emotional biases. I'm hidden. Can you see my panel here too? Can I get rid of it?
I don't see a panel.
Okay, good. That's good. I got it out of the way. In other words, relatively persistent traits and habits is what we're talking about here. All right. Now, many people, and probably we as cognitive scientists, find questions about the origins of these kinds of differences between people intrinsically interesting. But from a biomedical perspective, the incentive to study them comes from the very well-documented observation that many seriously adverse outcomes evolve disproportionately in the context of specific behavioral phenotypes. And among these are the following outcomes. Academic disengagement develops in a particular behavioral phenotypes, social dysfunction, mood disorders even psychotic disorders, and drug and alcohol dependence. But again, whatever their origin, one thing is certain: behavioral phenotypes don't just emerge suddenly at a particular point in time, but more gradually through cumulative effects of factors influencing them over the life of the individual. So these plots of data from my laboratory show that both calculation skills, which are clearly dependent on instruction and practice, and perhaps more surprisingly, even this basic measure of motor inhibitory function on the right, and I think I'm using my cursor on the wrong screen, over here on the right, show a very protracted period of development in children. Here you see the calculation goes up from age 6, at 16, it's leveling off, but it's still going up, just basic calculation. This inhibitory threshold, which is better the lower it is continuing to go down. You can see here across the whole age range from 6 to 16. But the question of central interest to us has been why these individuals at age 14, shown here, have calculation skills more typical of 9-year-olds, and these 9-year-olds have inhibitory function, which is more typical of 15-year-olds, right? So what do we know that constrains our models of the origins of these kinds of differences? Well, we know that when they have been studied with sort of genetic epidemiology, mostly, they all exhibit moderate heritability. But the genetic architecture appears to be highly polygenic. And so it's been difficult to decide what factors mediate this genotype to phenotype mapping, this heritability of behavioral phenotypes, this tendency for behavioral phenotypes to be associated with genetic factors. And what accounts for the large amount of the variability that is not, statistically at least, explained by common genetic variation, which we can model now with genomics and with family studies. Well, how do we build and test a model of a developing behavioral phenotype? Well, in our group, we think of we have a kind of conceptual model of the origins of behavioral phenotypes as a function of first the neural genotype. Now, this is hypothetical. We don't know what this is, but presumably there is a set of factors in the genome which influence some neural parameters that differ among us. And in some cases, effects of environmental factors that directly affect the biology of the brain. And what I'm talking about here is things like perinatal damage, for example, to the brain that might influence a child's development over a long period, because this is an environmental impact, but it would affect the child's brain function. But these cases are maybe toxic exposure of something. But much more generally, through the cumulative experiences encountering and interacting with the physical and the sociocultural environment. And importantly, what we now call the interplay between the genetic, the neurophysiological, and the environmental factors, which we refer to as either GXE, which is gene by environment interaction, or RGE, which is different. It's correlation between genotype and environment. So how do we achieve the kind of integration we need to improve models of such a complex process that is acting at so many different levels during the development of human beings? Some of us came to the conclusion, as I say, about 10 or 15 years ago, that we might start by observing the developing mind and brain closely and continuously. And so when the first stimulus package came around back in the last recession we had, we pulled together a group here at UCSD and took our first step, a major first step in this direction, with a project called PING, which stood for Pediatric Imaging, Neurocognition, and Genetics. It was funded by the American Recovery and Reinvestment Act at that time. It's probably before your time, but kind of like the big stimulus packages we're having now. And Anders Dale and I kind of organized a group, many other folks here in La Jolla that were our collaborators, and also 10 other distinguished institutions and many collaborators that formed previous collaborators and people that we recruited for this project together. And this project had only one specific aim, and that was to create a public pediatric imaging genomics database. And that's because at that time, there were some growing databases with brain imaging and genomics and some behavioral phenotyping in adults and particularly in aging adults, but there were none in the pediatric age range of any size. And so we worked together with these folks at there were 9 sites. And we had several cores. You see those here, and those cores were based here in UCSD. A number of our colleagues were involved in them, and some at Scripps Research Institute at that time. And then these were the different sites and our site colleagues. We rapidly, in a 2-year project, because this was a stimulus package project, and at that time, everything had to be very fast. You had to do everything very fast. You only had 2 years. You had to have a project that they considered to be shovel ready. So we convinced them that this was shovel ready. That was quite a feat. But anyway, we quickly recruited participants. These are typically developing young people between the ages of 3 and 20. We screened and initially consented 1,700 of them. And we got the key deliverables in over 1,200 of them. And the key deliverables were modest because, of course, this had to be done very quickly. Demographic, neuromedical history, family history data. We used the just minted NIH, National Institutes of Health Cognition Toolbox to assess neuropsychological functions and cognitive function. We measured some social emotional phenotypes in a subset with another stimulus package project, the Phoenix Rising project, also sponsored by the NIH. And we did multimodal brain imaging. These are probably, they may be familiar to some of you, but they're basically structural imaging and functional imaging. And then we did genome-wide genotyping. And in the interest of time, I'm just going to touch on one set of observations that we made with the Ping resource. It's been a while ago, but although the sample was cross-sectional, we just, as I said, we just had one observation in kids across the age range from 3 to 20. We were able to establish unequivocally that the surface area of the cerebral cortex continues to expand until it peaks at approximately 10 years of age, and that it then begins to contract subtly, but continuously thereafter. And more interesting than that even was that they were able to dig down under this global effect and estimate the effects at each vertex on the tessellated surface of the cortex with the best methods available, which were, by the way, developed by Anders Dale, our colleague, who was a CogSci, one of the early CogSci PhDs, who went off to Harvard and was at MGH for a while with an imaging group there, and then came back to UCSD and is now professor and vice chair of radiology and also professor of neurosciences, and my partner in these projects. Anyway, so so you can see that what we're able to do here is to actually plot for every point on the vertex, on the surface of the cortex, an estimate of the annualized rate of expansion in the warm colors or contraction in the cool colors over here. And you can see that at ages 4 and 6, if you look at this, this bar right here, that in all these areas that are yellow, the annualized rate of change in cortical surface area is in excess of positive 3.5%. So 3.5% increase in surface area at each of those yellow points. At ages 4 and still at ages 6 in a little bit smaller areas here, and a little bit, and every point on the cortex is expanding. In the red areas, just not quite as fast as in the yellow areas. And now we're going to change to a different, the rate of change slows down a little bit, and so we're going to be looking at this bar over here to estimate how much is changing. But you can see that at age 8, it still is the case that that at every vertex on the cortex, this is just showing you all the different views of the cortex so you can see it all. The cortex is expanding in the yellow areas, again, in excess of 2% a year. In the red areas, just a little bit, more in the 1% range. And then something funny happens. At age 10, it's almost a wash. All these annualized rates of change here are very close to 0. And that's why they're a little red or a little blue or gray. By age 12, you can see that virtually every point on the cortex is beginning to contract in area. And you can see that even more clearly at 16. And this continues pretty much continuously afterwards. But one thing I'll just point out to you, because I'm going to return to it later. At age 10, we can see that things are turning around here because we have the whole arc here. But it's a little hard to tell whether the differences between these redder and bluer areas are really different from each other, or if this is just kind of in the noise. But that was with 1,109 Ping participants aged 3 to 20. So in summary, Ping has contributed to a more powerful data-driven set of analyses of genetic, imaging, and behavioral measures to model cognition and brain development. And there have been over 100 publications that have used Ping data, which is now publicly available because we shared it with the entire scientific community through the NIMH Data Archive at the end of our project. And so it is now available there for scientists to put together with other data to make even larger data sets and so on. But it was a cross-sectional design and there were very limited assessments. And so this limited the power of the study to really explain these developing individual differences that we're so interested in. And frankly, the cohort characteristics did not very closely resemble the population, the US population, for example, because it was kind of a convenient sample. But Ping definitely helped to lay the groundwork for the longitudinal multi-site study, the adolescent brain and cognitive development study, which I'm going to talk about now. We call it the ABCD study. Okay, what was the rationale for it? Well, first, as you've seen, neuroimaging at the time that we began the ABCD study, after the Ping study, neuroimaging had always clearly revealed that there was ongoing, robust biological development of the human brain throughout childhood and into early adulthood. But people had begun to focus, especially, particularly at the National Institutes of Health, on adolescence as a pivotal time in the lives of young people. Under the best circumstances, it's a time when there's a lot of increase in knowledge and skill and creativity, but it's also a time when the earliest signs of serious mental disorders appear in some individuals. And it is definitely a time of increased risk-taking behaviors. And that can be a good thing and probably is a highly adaptive thing that happens in our species and its development, because you need to take some risks to get out of your nuclear family, to go out into the world, network with your peers, find a mate, many important things that you need to do that involve a little bit of risk-taking, you know. But of course, it can, in some young people, get them into trouble because of accidents that may be pretty serious accidents that may lead to long-lasting consequences. And it can get people into trouble if they experiment too freely and with dangerous addictive substances and other habits that can begin to erode the quality of their lives. Okay, so at the time of ABCD, it was well understood in the scientific community that studies human development and in the National Institutes of Health and other federal agencies responsible for research on this topic, that health, mental health, social, and academic outcomes had been associated with genetic, environmental, and behavioral factors. But it was also clear that more effective preventative interventions of adverse outcomes would depend upon better models of the roles of these factors. For example, which ones were more likely to be causal, which ones were kind of mediated by their association with the other ones, and things like that. But unfortunately, the limited scope and size and sometimes the method sensitivity of previous studies had really reduced the impact of the research to answer these important questions. And I'll talk more about that later, but that was part of the rationale for ABCD. Now, the first idea for this large-scale study was originated at the National Institute of Drug Abuse and an entity, a collaborative research entity called CRAN. And this is a collaboration between the National Institute of Drug Abuse, the National Institute of Alcohol and Alcohol Abuse, and the National Cancer Institute. So it's really about, it's collaborative on addiction. And so that's really where the initial idea came from and where the leadership came from in bringing this idea forward of having a large-scale study of adolescence. But as the planning for the ABCD study progressed, it gathered support from a lot of other federal collaborators who began to recognize the potential for answering questions that are important to many other kinds of health and mental health and so on. And so ABCD is supported by 10 institutes and centers of the National Institutes of Health and 4 additional federal partners, the National Science Foundation, the Centers for Disease Control, the National Institute of Justice, and the National Endowment for the Arts. Okay. So what are the primary aims of this study? Well, there are a number, and as you can imagine, as more institutes began to get involved and provide some additional support and get interested in ABCD, the aims grew broader and broader. There's hopes for national standards for normal brain development across this important period in development. We are measuring a lot of individual developmental trajectories of brain, cognitive, and emotional development, also academic progress to some extent. And we are identifying factors that may influence these trajectories, either protectively or adversely. We're examining the roles of genetic versus environmental factors, and as I mentioned, their interplay. We're studying the effects of health, physical activity, sleep, and other variables on brain development and other outcomes. And because of a very large sample size, which I'll describe in a minute, we will witness and study the onset and progression of some mental disorders that have their onset during this period in development. We'll look at what seems to be related to the variability and the course or severity of these mental disorders and the relationship between mental disorders and substance use. And we'll come back to where we started with the National Institute of Drug Abuse, their interests, which are how substance use patterns affect developmental outcomes and vice versa. Okay, so this is the entire ABCD study. As you can see, there are 21 esteemed institutions around the country involved as research sites. Here at UCSD, we have the coordinating center and the data analysis and informatics center. Sandra Brown, who's the Vice Chancellor for Research here at UCSD, and I are the PIs of this coordinating center. We are the consortium coordinators. Anders Dale is the Director of the Data Analysis and Informatics Center. And one of the research sites, data collection sites, is also here at UCSD. That's that one right there. Okay, and this is the national scope of the study. You just see that there are these pins in all the different places where we are following young people. We had very well-defined targets set for us by epidemiology consultants to try to recruit a much more diverse cohort than had generally been recruited before in any research project of this scale. And so we recruited 11,880 kids at ages 9 and 10. And this is just showing some of the demographic characteristics of the general pop. We had two different cohorts, sub-cohorts, so to speak. One was a general population cohort that was primarily recruited through schools. And there are 10,150 of them, and this is the breakdown of their ethnicity. And this is the inside. This is what we actually recruited, and inside was our target. So we came pretty close, 97.4% agreement between our target composition and what we were able to recruit. And then we had an embedded group of twins who were recruited differently through a twin registry. Okay, the visit schedule is comprehensive assessments at baseline and at biennial follow-up visits. That's every 2 years, which include imaging and lots of other things. Briefer assessments face-to-face under normal circumstances in the interim year visits, and more frequent phone and web assessments. This is just a little summary of just to show you the high dimensionality of the study. We collect a lot of information with our assessment battery. This is what we collect directly from the youth. This just happens to be the baseline, but it gives you a good idea of what kind of things we measure. Physical health, mental health, neurocognition, cultural and environmental factors. We do look at substance use, obviously, in 9 and 10-year-olds. You don't get much substance use, but you find out other things about their risks. We collect informative biospecimens of various kinds, oral fluids for a number of things, most in DNA in the beginning, so that we could genotype the kids. Sometimes we collect blood samples, hair samples, baby teeth. We do a multimodality brain imaging paradigm with structural MRI and functional MRI, diffusion tensor imaging, if you know what that is. Several different tasks for the task fMRI and also resting state fMRI. And there are a number of other data sources that are more passive data sources where we link the geocoding of the children's environments where they live and have lived. We link that to external data sets so that we can figure out, for example, what kind of neighborhoods they're growing up in from public data available about that. And we take school records, we get Fitbit actigraphy from Fitbit and so on. And this is, of course, especially when the children are younger, we collect a fair amount of information from the parents, and that includes physical health, mental health, substance use, mostly in the parents, and culture and environment. Okay, and then just to highlight that we can't do this without our Data Analysis, Informatics, and Resources Center, which is Anders Dale directs. And I'll just go through this quickly just to show that they're responsible for getting the MRI, making sure that the MRI imaging that's being done at all 21 sites is well harmonized and is state of the art, and that we can get the same comparable data from all three of the main GEs, all three of the main vendors that make MRI machines, Siemens, GE, and Philips, because of course we have some sites that we have at least one site or two sites that have each of those. In order for this high throughput to keep up with this very large study, we employ fully automated computational workflows that are developed by Anders' lab. And the DARC also manages not only the electronic data capture and curation of our data centrally, but the public sharing of raw and derived data from all domains, and I'll talk more about that in just a minute. There are a lot of very fancy bells and whistles that our expert computational neuroimaging group provides. We do real-time motion correction, distortion correction. We use a very advanced diffusion imaging protocol for getting the most sensitivity to both intracellular and extracellular signals. And we, of course, they use all the information they collect, including phantom data, to calibrate all the imaging in all the modalities. And the end point is that we have over 20 imaging biomarkers that are measured within each of the structures, cortical regions. These are the structures, the cortical regions, and the major fiber tracts in the brain. We look at 20 different imaging biomarkers in each one of those regions of interest. And these are among tens of thousands of variables, tabulated measures in the ABCD data resource. So as you may have gathered, this is a study that is being conducted with a really aggressive open science model. And that means that as we're gathering this information in this longitudinal study where we're seeing these kids, pretty much all the time, on an annual basis, we release essentially all of the data that we have collected up till 3 months before that release. And for example, for the first release in 2018, there were 4,500 participants. The second release, all 11,800 of the participants were present for baseline data. And then there was some follow-up data. And in 2020, several thousand 2-year follow-up visits as well. Okay. So now I'm going to talk just a little bit about the findings that are emerging here, just to give you some idea what's happening with this study. And this is one of my slides that I borrowed from one of my graduate students. He says, brace yourselves statistically. But in this case, there's no statistics coming except to say, when you have the sample size this large, where you're up in the 10,000 range, whatever you do, you have power in your statistics. And so just to give you an idea, I just remember that when we looked at the ping data set with about 1,100 kids cross-sectionally, we couldn't really see much about, it was hard to say at age 10, whether really the whole cortex was just barely still expanding or was just barely beginning to contract, or if these regional differences were really significant. Were there actually some areas still expanding while other areas were actually already contracting? We couldn't really tell. We didn't have the statistical precision, but this is the same picture from the ABCD study, exactly that same window between 9 and 11 years of age. And here, these are actually highly significant, these differences. And so these are, we can see here that there are highly significant areas, contraction in some areas at the same time that there's still highly significant expansion in other areas shown in these warm colors. So this area in the pars opercularis and inferior frontal gyrus is expanding at the same time that areas in the occipital cortex, for example, are clearly contracting. Just more precision. So I'm just going to go over one example of some early work here, and I know we're almost out of time, but this is a work done by a wonderful postdoc who I'm lucky enough to have working with me and my research group, Claire Palmer, and she recently published this work in cerebral cortex. And we looked at we looked at the neurocognitive data, and we're interested in cortical morphology and how it relates to the neurocognitive data. And so she looked at two major indices of kind of cognition. Some people call it fluid cognition and crystallized cognition. Some people call it fluid and crystallized intelligence. It's really just cognitive test performance with either novel tasks or tasks that are more language and education related, crystallized. Crystallized being things like vocabulary and reading, oral reading, fluid being more like inhibitory tasks and working memory tasks and executive function tasks. And what we saw was that we looked at the pattern of regionalization of the cortex. That is, how much territory is given in a person's cortex to, say, the prefrontal cortex or the inferior frontal gyrus versus the occipital cortex and so on. And we can do that at a vertex-wise level at every point on the cortex. And so what we see here is the pattern of relative expansion or contraction, sort of the shape, if you will, of that cortex. It is most predictive of the fluid cognition measure. And this is the pattern for the crystallized cognition measure. And the important thing about the results is that the patterns associated with these two closely related phenotypes that are thought of as being strongly associated with each other are very distinct. And there were some things that we did to really pursue this more thoroughly by residualizing the measure, the fluid measure for the crystallized measure, and looking to see if that changed the pattern, and it didn't, and doing the opposite, residualizing the fluid with the crystallized, and it didn't make a difference. That's just what I'm saying right there. So the basic result was that each cognitive phenotype measured in the NIH toolbox is associated with a distinct pattern of cortical regionalization. So here you see, this is the crystallized measure that we were looking at, and the pattern that we saw associated with the scores, the variability on the scores of that. Here's the fluid composite that we were looking at. And you see this pattern is very different from this one. That's what I just showed you. This is a little bit easier to see it here. This panel compared to this one over here. These are the composites. You can see this is a very different pattern than this one is, right? And these are the different subtests from the battery that go into computing this composite. And these are the two subtests that go into computing this composite. And you can see that these are fairly similar to each other, but even where they're a little bit different, this is clearly a mixture, say a mixture of these two, you can see here. But in this case, it's even more clear that there are quite distinct patterns for each of the subtests, actually, this one being quite different from several of the other ones. But them having in common that they almost always have a little something here in this inferior frontal region. And so in the composite, you see that there. But the upshot is that the cortex of the best readers, for example, look at this reading test here, is not regionalized the same way as the cortex of the best cognitive set shifters, which is this one, or even the best encoders of a memory sequence, right, which is this one. In other words, different minds are associated with different brains. So why are these observations important? We think they're important because the results are consistent with a recent model of intellectual development that challenges conventional theories that a latent causal factor, which has been referred to as G, underlies variation in intelligence across all domains. This new model, called the Mutualism model, explains the psychometric hallmarks of G as a consequence of functional interactions between relatively distinct neural processing systems during brain development. And complemented by other work from our group, which examines heritability of these phenotypes, the results underscore the importance of determining how learning experiences of children with different neural characteristics may serve to amplify or suppress these positive interactions that are hypothesized to occur during development to give rise to these differences that develop between us in terms of what kind of, sort of how our minds work, what kinds of things do we take to, what kinds of strong skills do we develop. And so the point here is that this kind of underscores that we are all different from each other. In some ways, this is reflected in the differences in some of our neural characteristics. And it's also reflected in the kinds of things we do really well and the kinds of things we do just more average. There is a lot of variability, and it's very important for us to understand more about how the learning experiences of children with these different neural characteristics are giving rise to are influencing their creative development, intellectual development. And if we understand that, we'd be able to boost the positive interactions that occur and ensure that more kids develop high levels of creativity and resilience that really give them the best opportunity to develop their unique set of strengths, their unique architecture. So anyway, this is something I just want to chat with for just a minute because it's very important for us all to understand this. I mentioned before that one of the bullets in the rationale for the ABCD study was that the studies that had been done before were often too limited in scope and size, in terms of dimensionality and also in size. And that was it was an understatement, because there has been something of a crisis of replicability in our field, not just ours, in many parts of biomedical research and other kinds of research. But what do we mean by replicability? Well, replicability means that effects are similar if they're estimated in comparable but independent samples, right? That's kind of what we mean by replicability. I borrowed this slide from Wes Thompson, who's the chief biostatistician at ABCD, but I think we all understand that. And so what this graph is showing us is that let us say that we have actual effects of the effects of individual factors, for example, individual correlations that reflect real causal influences on a phenotype. Let's say that they're relatively small. Here's a row of 0.10. That's a small effect by most standards. But let's say that that's the real effect in the population. What happens when you have small underpowered studies, which is all of these that you see here, these are the sample sizes of previous studies, or the typical previous studies, and this is like 10 people in a sample, and then a little bit larger sample here, and then a little bit larger up here to closer to 100. What you see here is that these are the sampling distributions. If you're looking at these variables that are correlated with the row of 0.10, and you look at the sampling distribution, you will see that that there will be a very wide sampling distribution at these smaller sample sizes, and that most of them will be very inadequately powered to detect an effect of this size. But some of them will appear to find an effect that is larger than that, and only if it's considerably larger than that. See, some of these spurs, this is just like, these are obviously very inflated. If the real effect is 0.1 and you get a statistic that's up here at 0.5, then that is spuriously inflated, but it may need to be that high to be statistically significant with this sample size. So only those spuriously inflated effects that occur out there in the thousands of people who are looking at these two variables, looking for a correlation, will be published because we only publish positive effects. And if you have a lot of underpowered studies like this, you'll see a lot of effects entering the literature, which are really false positives, or they're highly inflated estimates. So there may be a small real effect, but they're mostly going to be very inflated estimates of those effects, because only those will exceed the criteria for statistical significance, which gets your result published. And so this has been happening in the literature for a long time. And because of the positive publication bias, compared with the fact that only spuriously inflated effects ever make it to the literature with small sample size, we have a very biased literature. And it exaggerates the effects. And in some cases, what we're finding is that some of these effects that have been reported don't replicate at all. They were entirely false positives. And this is something that is much better understood now. But what's the good thing about ABCD? This shows you that at the sample size of ABCD, not only can you detect very reliably these effects at only 0.10 here, but they are significantly different from 0. So you have a lot of power, and the confidence interval around that estimate is very small. So you really have much more precision to estimate these parameters with these very high sample size, especially in the situation where the effects are mostly small. So it's the case that early observations from ABCD are going to force corrections in many research areas where the sizes of the effects reported in the literature are likely to be revised downwards Some may be shown to be absent or really trivially small. And I just want to point out that I don't have time to talk to you about it yet, but this scenario resembles very much the period in human genetics when genome-wide association studies failed to replicate many candidate gene effects. It's a very similar situation. And we now know that that these effects, that really the gene effects that are real are all are mostly very small and all complex human phenotypes. The heritability of those phenotypes is the result of many variants in the genome, each exerting only a very small effect, but in aggregate, adding up to large effects on the phenotype. And so this is the shift that we think was coming from ABCD in general is very similar to what happened in human genetics about a decade or so before we had a lot of large-scale GWAS studies to help us figure out what was going wrong and get it right. So it's, we should take solace from the rebirth and excitement that the human genetics field is now enjoying as it gets on with the challenge of associating polygenicity with the complex interplay between genetic variation and environmental factors. And we're going to do the same thing with ABCD. So I don't think I'm going to go through this last slide. I'd rather stop and let you guys ask me questions. It's not really important anyway.
So about the replicability thing, so do you think like the field's going to change a lot because of the findings?
Yeah, I think it's already changing a lot because we actually will have, from these large population-based studies, and because we have so many, we're measuring so many things, and you can see in our assessment battery. I'm going to just stop sharing so I can concentrate on you guys. I don't think I need my slides anymore anyway. Yes, that's better. So because we have so many different kinds of, we have lots of environmental measures, we have lots and lots of things that might influence kids' development. I think what we're going to start to see is that people kind of give up on, I hope, on certain kind of silver bullet explanations that that people think, well, I know why some kids can't read. It's because they didn't hear enough words in their infancy. That's a very popular one right now. I mean, I'm exaggerating a little bit, but I know, and there are observations in the literature that would suggest that this is a very large effect. Problem is, there are lots of observations like this, and we're learning more about how they might enter the literature. It's not that anyone is doing anything wrong or that there's any chicanery, nothing nefarious going on here. Just that the way our publication policies, the way we do the structure of the research, it's supposed to be hypothesis-based. You make a hypothesis. Lots of people are asking that hypothesis all over the country, all over the world, in fact. And they're all controlling their false positive rate at like 5% or something like that. But that just means that if you're doing hundreds of them, then 5% of those things that seem to be significant to the individual investigator, and they think they can reject the null hypothesis, they're actually false positives, because if you just do it everywhere, that's what that means. That's kind of what the sampling distribution is. And so those papers get reported, and if they were large enough, the observations, the statistics were large enough to reject the null hypothesis, they're at the very least very large inflation of the actual effect sizes, because that's the only way you could get such a thing in a small sample. So it's really, it's a bias in the literature. So people will be able to check out some of those things with these much smaller error bars around those parameters. And we'll be able to see that those effects may be present, but they're probably much smaller if they are. And they may actually not be there, especially if you look to see what other kinds of factors that are sometimes conflated with them might actually be mediating them, right? So, you could have something where, for example, that you're, you think that it's something about a child's experience that's in the home that is something about having books in the house or something like that that causes a child to have more trouble reading later on. But it may be that living in that house is also a place where there's more air pollution, or there's more asthma, or there's more illness or something like that. And that it is maybe that association that has been observed between the number of books in the house and reading is really mediated by the other correlates of poverty. Maybe it's living in a neighborhood that's not very safe, or the stress associated with having, food insecurity or whatever. But those variables haven't always been available to look at in a model that allows you to see, well, let's see if we model this with, with very large sample size and with measures of all of those things that could be kind of related to each other, and so could be kind of confounded, and do we still see evidence for an independent effect of how many books are in the home on later reading performance? Or is there really nothing related to books in the home per se, after you account for the level of poverty, the stressful life events in the parents' lives, or whatever? I'm just making this up, but you see the point that if you can model these effects with a lot more of the other variables that could easily be confounded with these your favorite variables, you get a better idea whether which ones really show an independent relationship. It's an observational study, so an observational study cannot establish causality, no matter what anybody tells you. There are fancy statistical methods that people say builds a stronger case, but basically observational studies do not prove causality, but they can allow you to model outcomes like reading ability, which is something we're doing a lot of right now. What differences in reading ability in 9 and 10-year-olds, right? You can model it with a lot of different variables that might influence it simultaneously to see which ones remain plausible, more potent predictors, even in the same model, even when they're in the same model, with measures that could be confounded with them in many cases. And of course, sometimes when they're completely confounded, there's no way of knowing but at least then that you can't rule out that the effect that you thought was going to be a large effect on reading, individual differences in reading, was going to be due to this variable that really is so confounded with other variables that there's either no case to be made for any of them, or some other measure appears to be mediating fully that apparent correlation with the variable that is your favorite variable. So it's really, I think the way we should think of it is that human development is extraordinarily complex. And all of those things that we talked about, who you are constitutionally, the genes you inherited from your parents, that influences who you are to some extent in subtle and interesting, very complicated ways. But of course, when we're talking about mental phenotypes or behavioral phenotypes, your experiences in the environments you're in are enormously important in who you are. What you learn, who you encounter, where you go, all those things, very, very important in who you are behaviorally. So all of those things are important in who people are, but that's very, very complicated. So if you're going to learn to understand it better, you need to measure you have to measure a lot of dimensions that could be influencing how you're developing, your habits, your traits, your experiences, and so on. And you have to have a sufficiently robust corpus of data that allows you to get reasonably stable estimates of all the parameters in that complicated model that you're trying to build to understand human development better. I think of it as being very much like cosmology. That's my favorite analogy for it. Because the developing human mind and brain is approximately as complicated as the dynamically changing universe, known universe. And so why do we know so much about how is it that we, these little insignificant species on a relatively unremarkable planet on a completely nondescript galaxy in a completely nondescript galaxy. How do we know so much about the structure of the universe around us? And even what is going on in some of the dynamics in the universe? How do we know that? It's not because we're just so clever. I mean, it is in part because we're so clever, but it's not really, it's because of the enormous data resources that have been amassed over many decades now, taking every signal that we can possibly take from the heavens and building these huge cosmological databases. And all of the hypotheses about all of our knowledge of the structure of the universe has to fit, has to be consistent with this constantly increasing data resource, trying to collect signals, as many signals about anything happening in the universe as we can, and recording those observations for scientists to use and to extract structure, meaning, dynamic parameters, and so on from. And so to me, it's a similar thing, is that we, in order to move past, sort of simple, but not actually very useful models of such a complicated thing, we have to build data resources that are up to the job. We have to do that collaboratively, because this is a highly transdisciplinary. There are at least 200 leading investigators in the ABCD study, and we are all working alongside each other every day just to run this study. And we come from all different disciplines, all different kinds of training. Well, not all, but many, many relevant disciplines. And we work on it together because we know that we are turning the corner with this new paradigm, a population neuroscience paradigm. It is what we need to understand something as complex and subtle as the human mind and brain and how it develops. And of course, it's just at one level. It's just at the level of, of things that we can measure in vivo. A lot of basic neuroscience going on elsewhere that will also inform this endeavor. So anyway, I'm talking too much, but I do think it's going to I think the paradigm is going to absolutely transform how we think about ourselves and human development.
So, go ahead.
Sorry. I was going to ask, when you say the brain surface area contracts and expands, does it mean like the volume of the brain is contracting and expanding, or does it just mean the brain is becoming more wrinkled or smooth?
Well, it actually, it's a good question. There are some subtle volume changes in the brain, and it is the case. So in brain development, the brain tissues themselves are almost from the very beginning going through these waves of progressive changes. And by that, I mean things that add volume. And almost at the same time, right behind that, regressive changes that take away volume. So we know, for example, that many, many, many more neurons are produced in the brain than ever survive and are retained. Many, many more glial cells are produced than survive and are retained. Many, many more synapses are created than survive and are retained. But if you look at the whole thing happening in its dynamic, very, in different parts of the brain at different times and so on, it's also what we call heterochronous. It's like certain parts of the brain develop earlier, some later. So there are these. So what you really see is these waves of things that involve brain growth, but they're kind of going on in parallel with some things that are already beginning to be regressive. So the short answer is that at about age 10, up to age 10, the progressive processes are winning, if you will. There are some regressive things happening all along, but there's so much more that's happening in the brain that is progressive, that is adding cellular structures, adding cells, increasing the size of cells, and so on, that dominates. And so you see overall brain growth right through the fetal period, of course, and postnatally, until about age 10. And then the volume of the brain becomes stable, or if you pay close attention, slightly begins to reduce. Not because the development is complete, but because the progressive changes have now kind of given way to the continuing regressive changes. So it's the balance between the things that are progressive going on in the brain and the things that are regressive going on in the brain that determines the brain's volume. And so at about age 10, the balance tips to the regressive side. And when we're looking at these imaging phenotypes, we can measure the cortical surface area quite well. So we can tell you there's a cortical surface area, which is what I was telling you about. It's kind of what it sounds like. It is the cortical surface area, and it does expand until about age 10, and then it begins to contract. And you can see there's a pattern to it, which we were able to confirm is a real pattern with ABCD for sure. But you can see it with the ping data that I showed you with the yellow and the little spots. So the surface area definitely it expands continuously until about age 10, and then it begins to very gradually contract. That we know. We look at the thickness of the cortex, and that's different. We can measure the thickness of the cortex with imaging, but it's what we call the apparent cortical thickness. Because what we're really looking at is really the contrast between this essentially unmyelinated cortex, a 6-layered structure that is essentially unmyelinated, except for some myelinated fibers, particularly down in layer 6, and the underlying myelinated white matter. That's what produces the contrast on MRI that allows us to look at the cortical thickness. But of course, there's dynamic changes still going on, and we know now they go on for quite a long time, where even the myelination of the white matter is continuing over a very long protracted period of time. So that can shift the apparent border between the unmyelinated gray matter, which has a different signal value on MRI, and the myelinated white matter underneath it, which may be being myelinated quite late fully. So that shifts the boundary a little bit. So some of what we're looking at there is really probably not actual shrinkage of the thickness of the cortex, but the final stages of myelination in the deepest layer of the cortex and or the just subcortical U fibers that connect the nearest parts of the cortex. So, so we're still learning about that. I thought you were hoping for a simpler answer than this, I'm sure, but it's kind of more complicated than that a little bit. It depends on what compartment you're talking about, but the overall volume of the brain, it increases about age 10. Then it begins to very gradually reduce a little bit. And you can see that because there's more cerebrospinal fluid around the brain, because that reduction of the volume of the brain is accompanied by a little bit of increase in the cerebrospinal fluid around the brain. And of course, in old age, that gets very pronounced because the brain begins to shrink more dramatically. And almost all older people, my age, I'm sure it started, I don't even want to know. But there, there you can see this very pronounced change in the cerebrospinal fluid around the brain, because the brain's tissue itself has begun to shrink, mostly in the white matter. Anyway, that's the answer to that question, I think.
Thank you.
I hope it was at least partly helpful.
Yeah, it was.
So you talked about the replication crisis there at the end, and you made some great points about what can be done on the side of both the researcher and the person consuming research to try and account for that. But do you have any thoughts about what academic journals can do to incentivize, more research that's more in line with, good science? And do you actually have any examples of journals that are making steps to go in that direction?
Well, I think it is still settling out. So I don't, there isn't much consensus yet about this, I think, but I think it depends a little bit on the field. So in genetics, the journals have become extremely conservative. So they, you simply cannot get what we used to call, what we called a candidate gene study published anymore. You can't look at a particular variant and say, this was my hypothesis. I believe this variant is going to be related to this illness, or it's going to be related to this phenotype. And I'm going to go in there and I'm going to collect a bunch of people. I'm going to genotype them. I'm going to measure that phenotype, and I'm going to show that this variant that's in this gene, that, for example, the gene that codes for the dopamine transporter, and it's going to really affect this dopaminergic related symptom that some people have. They can't get that published because it's just not considered trustworthy. You know, if you just do a single test and you're looking for an effect that you could observe with a small sample, or even a moderate sample, you have to. They've kind of gone overboard in my mind a little bit. It turns out that there are considered to be about 1 million independent tests that are conducted when you do a genome-wide association test. That's looking at all the variants all the common variants that are known to exist in the genome. There are about 1 million tests in that. And that's what's done now to look for genes that are related to phenotypes. You do a genome-wide association study. But to avoid these false positives, you have to have your observation has to have a p which is smaller than 5 times 10 to the minus 8. That's many zeros. 0.00000005.
7 or 8, right?
So it's a lot. So that's a very conservative test for any given variant. But most journals that do genetics now, even if you have a very good reason to, you're only going to look at a certain set of SNPs which have already been selected, pre-selected or something like that, and you've got a very good case there, it's hard to get them published because they're so conservative that almost every gene variant association with a phenotype now has to be significant at that level. And of course, the only way you can really do that is to work with these really huge sample sizes. They're essentially meta-analysis of genome-wide association studies. And for example, the trait height, that is highly heritable, 0.9 probably on average. 90% of your height is, well, I won't say it that way. In a population, about 9% of the variability is related to genetic factors, additive genetic factors. Probably. That may be a little high, but that's an estimate from twin studies. But in that, there are now studies with, more than 1 million people in the study that are put together, because height is so frequently measured in a genome-wide association study of anything, whether it's in psychiatric disorders or cardiovascular disorders or blood pressure or whatever. So you can take that data and you can look at the association with height, and you can put it all together, and then you can find it. You can find the heritability for height, but it turns out that heritability is a whole lot of variants, many variants in the genome, spread all across the genome, each exerting only a very small effect, but additively adding up to pretty close to that total heritability that's been estimated in twin studies. So that's the way it looks. And so that's how people, that's why you pretty much have to take these very large studies to get anything published in human genetics these days. Now, there are some new methods, though, that allow you to really exploit those large data resources to ask a lot of interesting questions about the genetic architecture of individual phenotypes, but also the genetic architecture that is sometimes shared across disorders. So a kind of what we call pleiotropy, sort of the evidence that some genetic factors may both predispose you for bipolar disorder and also be associated with an increased risk for schizophrenia, for example. Some part of the genetic architecture would be shared in those two cases. Some other part would be different. And so those kinds of studies are developing now. And so now people have moved on to using these large data resources, identifying these variants in the genome, figuring out which genes they're in, using very large ontologies that have linked all these gene products to individual biological pathways, And so you can go and you can take your results from a large GWAS analysis, and you can go and figure out which biological pathways these variants act in. And then try to begin to pursue a more biological model from that of how the genotype to phenotype mapping is happening. But it's it's very complicated. And it's sort of like when you look at it with hindsight, hindsight is 20/20, but it's like, of course it works that way. It's sort of all, it makes sense. It's not convenient, but it makes sense that that's the way it works. But it was really, in genetics in particular, it was really just devastating when the first GWAS results came out. And these searches of the genome to find the sources of heritability of heritable diseases, for example, were just appeared to be a dismal failure because it was so hard to identify. Even a small percentage, to account for even a small percentage of the variability with the genome-wide associated hits, sort of GWAS hits, so to speak. So it's taken a complete change of conceptualization of how these things work. And frankly, I think that's kind of what we're seeing in our field too. There's so many. It's such a complex, dynamic process by which a person's personality, habits. I'm particularly interested in the development of intellectual development. I'm kind of interested in how people find the things that they're interested in, become engaged in them, develop a lot of creativity in those domains, develop a lot of expertise in those domains, those kinds of habits of mind that lead to resilience, to lead to self-efficacy, and pursuing whatever it is that a person pursues, right, in a way that's healthy and gives them more independence and more resilience. I'm really interested in that, and I think it's it's partly, we call it the development of creativity or the development of intellectual ability, but it's not really. It's really just the whole person developing in some way that gives them kind of skills and habits that are sustaining for them, healthy for them, and sustaining for them, and make them happy. And so I think that this is a very subtle and complex process, and we shouldn't be naive about it. It's going to be complicated. But I think the insight from our work that we think is very important is that some people like to think that it's, people are blank slates and it's all about what happens to you. And I don't think that's true at all. I think it's a very strong interaction between who you are constitutionally and then what happens. And I think we do a great disservice to many people in our society if we think it's fair to treat everybody the same, that everybody will benefit from the same experiences. I don't think that's true. And I think if we get better at this, it's we could realize enormous increases in the realization of the intellectual and creative potential of many people who are not being served well by our current methods of helping people, nurturing their development. So that's why I think it's so important to do it.
Cool, thanks.
Well, thank you guys for hanging out. I can see you've been very polite, and I'm sorry to have gone on a little too long.
That's okay. It was an interesting talk. So do you have any parting advice for people, maybe students who maybe want to get into that research? Or
well, yeah, there's so many ways to get into it because, as I say, there's so many different people with different kinds of training working on it. So it really, it depends a lot on what you find interesting, what kind of things, part of it you might find interesting. But in terms of, just becoming somebody doing cognitive neuroscience or developmental cognitive neuroscience or just cognitive neuroscience at all, I know that it is already kind of challenging for people because there's so many different programs where you can actually learn some of the relevant stuff in cognitive neuroscience. And it's hard to know which way to go, right? There are these, first of all, there's all these different behavioral science programs that call themselves different things: cognitive science, psychology, cognitive neuroscience, cognitive science. Oh yeah, there's that one. How do you know which is which one's the right one for you? It's very challenging. I can really appreciate that. And of course, then there's the whole medical way to go. You can go into, you can do medical training and you can become a neurologist or a psychiatrist. And the thing that people don't often know at UCSD, which always surprised me, is the way I went, which is to go into a clinical, in my case, a clinical neuropsychology program. Actually, we built a clinical neuropsychology program here. So that's another way to go, where you do get clinical credentials, but and you have access to clinical material if you want to study a, patient population or something like that. But so it depends on what kind of things you're interested in studying. But that's another thing to consider, which has pluses and minuses and so on. So, but it's really, I think, first of all, make sure you explore the different things. But I think if you can spend some time working in research, sometimes after you graduate, that's often very helpful. I think a lot of people figure out what part of this they like, what kind of doing research like this do they like, and what parts they don't like. Actually working in a research lab, if you can do that for a while, pick something that you think sounds interesting to you, that you have an opportunity to get more experience in, and from that vantage point, talk to other people, maybe other people who are in the lab who are trying to figure out which graduate program they want to go to or what kind of research they want to do, but also the faculty that you have access to in that situation. That's one way of kind of giving yourself a chance to get a sense of which way to go. But also, just try to pay attention to what's going on out there that you think is really interesting and talk to the people who are doing that, figure out what they're trying next. Maybe go to a department where they are. I think that's maybe a helpful thing to do. I didn't do any of that stuff, so it's hard for me to say those things because there wasn't a place to go. I was just in the right place at the right time, basically. But I can see that from all the students I've worked with over the years that finding something that seems like it's attracting you and then spending some time there. I think it's such a big decision that you make when you go into graduate school and you kind of get paired with an advisor and you're in a department and so on. Spending a little time in the research world in between your bachelor's degree and your entry to graduate school, if you're considering going to grad school, it's not, I think it's a worthwhile thing to do if you can. I know not everybody can, but because it gives you a different perspective, gives you a little more time to look at things from a different perspective than you do as an undergraduate, where you're focused on graduating and getting degree requirements done and stuff, and haven't had a chance to look around as much at what research is out there and talk to academics on a more daily basis about where they think the fields are going and so on. I guess that would be my one bit of advice. Give yourself a little bit of time to make an important decision. But the other thing is that, careers are really long. I mean, I have reinvented my research programs so many times, and I've done so many different things at different junctures. So people, and you could always, people, you should never be afraid to do a reset if you feel like, okay, I already spent a whole year in this program, but I think I finally know what I want to do, and it's over there. I would say go for it. Because it seems like it's really critical that you get that degree and get on with your life and, make more than a subsistence income and all that stuff. I understand that, but it's a long career ahead of you. And so from that perspective, on the other end of that very long career, I can tell you that, ending up where you wanted to be is more important in the long run. So don't hesitate to make a pivot if you feel a strong pull somewhere else.
All right, thank you.
Sorry, can I ask one last question?
Sure.
How did you personally pivot from social sciences? I think you mentioned doing that for your BA into what you do today.
Well, as I say, I ended up at Irvine, and it was the only program that they didn't have a psychology department. I started at University of Texas at Austin. At 17, I left home in Mobile, Alabama, and went to University of Texas at Austin as an undergraduate. And this was like, like my whole life, my whole world opened up. I'd never been in an academic setting or anything like that. And and I went into psychology because I was interested. I was interested in these phenomena from an early age. Not in this way. It wasn't so much the biology or the neuro part of it, but just people and why they were different and people's minds and how their feelings and all that stuff. And so I went into a conventional, traditional psychology department in Austin, but I went to Southern California, and I moved to Southern California, and I wanted to go to UC Irvine, but they didn't have a psychology department. They had something called social sciences, but it was really kind of quantitative social science, if you will. There were people studying vision with computational models of vision, for example, and economic behavior with quantitative models of economics behavior, human behavior, and and people who were studying psychoacoustics, with auditory processes and things like that. So it was a very different kind of department, but it was very technical. It was more quantitative, and there was a strong psychobiology department there. And I kind of got wind of some of that. There was some stuff going on in Boston at that time. That was kind of behavioral neurology, but there were a group of people who were trained as psychologists who were beginning to do this stuff, which they were calling clinical neuropsychology. And I was fascinated by that. I was also fascinated, by the way, by these early books by what's his name? Guy who's just this is my old memory now, but the woman, the man who mistook his wife for a hat or something like that. Anyway, these really interesting behavioral neurology books. And so I was interested in those things even when I was still an undergraduate kind of behavioral sciences student. And that's why I said I was vacillating between getting more training in neurobiology and really becoming more of a neuroscientist, or going the way I had been going, which is to be a clinical psychologist. But as I mentioned, I kept being interested in the brain. This is before we had brain imaging, so you have to remember that. So I was still interested in the brain, and people were studying the brain by looking at people had different kinds of brain lesions or different brain disorders, and then trying to figure out how that related to the symptoms they had. And that was what clinical neuropsychology was. You did a bunch of tests that tried to pinpoint where you thought in the brain their lesion might be that would produce these symptoms, this profile of symptoms. And I was intrigued by that. And so I wanted to learn more about that. And that's why I went to Palo Alto. That's why my director at UCLA, let me go up to Palo Alto because they were doing some training of clinical psychology trainees there. And so I got some of my training there. And that was when I discovered this machine that was in the basement, this prototype CT scan machine. And of course that changes everything, right? Because you don't have to now do a bunch of tests. You actually can see where the lesion is in the brain. And that just blew me away. And I just happened to be able to program a computer, and I was able to figure out how to measure quantitatively the characteristics of the brain tissues from CT scans. That's how I started. And then I put that together with the cognitive testing and this new field that was just being created, neuropsychology, even for clinical psychologists, and also kind of cognitive neuroscience, what would become cognitive neuroscience, was being born. And I was off to the races. It was just a very exciting thing. And I made the transition to MRI when I came here to UCSD and started doing quantitative MRI. And was hooked on that. So I grew with the new field, so to speak. So I don't know if that's your answer, because it really is kind of, being at some place at the very beginning when it became possible to do this, and then recognizing this had to be the way to go. Not that it's answered all of our questions. We've been a little bit overly exuberant about the brain imaging results, to be honest, for much of the time, because it's still fiendishly complicated, and it still doesn't tell us all the answers, by far from it. But it's still very exciting to be able to look at, you know, the human brain and how it's different in different people and relate that to their mental functions and so on. So that's what cooked me. Maps.
Thank you. That's really informative.
Okay, thank you. But you know, there's so much happening for you guys. I mean, the thing is, the specifics don't matter. You know, it's the field of, you know, our field is expanding very rapidly. Lots of interesting things happening now. I think it's going to be a very exciting time to be in this field generally. And it's going to take over a lot. There's going to be ways of doing it in many different disciplines, because it's going to be a very highly transdisciplinary enterprise, very collaborative. And so, you know, there are going to be these opportunities that come along. If you're paying attention to what's happening, there are going to be these opportunities that come along that are just as exciting as the ones that came along in my time, that I was able to kind of go, okay, I'm going to try that. I'm going to see if I can do that. And so they're all out there. All these, it's not the same thing, but it's a very dynamic time. There's a lot happening in the field. The field is growing always larger because there's so many little niches within there where there's a lot of interesting work to be done. And as we accumulate these, if old people like me just keep giving you guys lots more data, you'll have a much more powerful observational base. You know, like you can become the cosmologist of the brain in the future, because you'll have those data resources that you can use your creativity to try to make sense of. And I think really that's what we're trying to give you with these large population paradigms, is more useful observational data to be used by clever young scientists of now and the future to begin to extract the most meaningful models of these complicated phenomena, and to really find the signals that are actionable, that will help us to improve the outcomes, reduce the adverse outcomes, and boost the well-being in more people in our societies. You know, we have a long way to go as a species to do this right. And so I think it's a very exciting time. There'll be lots of opportunities. Keep your eyes open.
All right. Thank you very much for coming and talking.
My pleasure, always. You guys have a good evening.
Yeah, you too.
Bye now.
Bye.
CSSG secret meeting time.